Hi everyone, welcome back to Neurobiology at Providence College. I'm Joe DeGeorgis. Today we're going to talk about a very interesting paper. This is our third lecture for Exam 3 of PC Neurobiology, spring of 2020 in our online format. The paper that we're going to talk about is titled Fatal Familial Insomnia, a prion disease with a mutation at codon 178 of the prion protein gene, by Medori and colleagues. You know from reading the book, The Family That Couldn't Sleep, that of course fatal familial insomnia is a sleeping disorder, and they say that it's characterized by marked reduction or loss of slow wave and rapid eye movement. In more advanced stages, the patients have complex hallucinations, followed in the terminal stages by stupor and coma, and then eventually death. They say that the average age of onset of the disease is 49 years with a mean duration of 13 months. So, of course, it sounds terrible. You stay awake for 13 months and then pass away from this terrible disease. They also knew from autopsy that at least one individual in the family had spongiosis of the cerebral cortex, which of course is known to be a hallmark of Kuru and Creutzfeldt-Jacobs disease. And I think at this time they knew that Creutzfeldt-Jacobs both had a sporadic form and a heritable form, that is a form that's caused by a genetic mutation. So not unlike Alzheimer's disease. And of course, Stanley Prusiner had argued emphatically that Kuru and scrapie, mad cow disease, Creutzfeldt-Jacobs, and so on are all caused by a protein, not a slow-acting virus, not DNA or RNA, and he named that protein prion. In the paper, the authors first show the pedigree of this large Italian family and the individuals that are afflicted with fatal familial insomnia. And they say that the disease is inherited in an autosomal dominant fashion. We talked about this when we were looking at mutations in APP and related to Alzheimer's disease. And the argument for why that is thought to be an autosomal dominant disorder is the same as the argument here. First of all, we know that it's not recessive because the disease does not skip a generation. So it's not recessive. It can't be Y-linked because females have the disease. It can't be X-linked dominant arising from the mother because any son that's afflicted with the disease must have a mother who's afflicted. And it can't be X-linked dominant from the father because all daughters of an afflicted male must be afflicted. Okay, so if you stare at this long enough, you realize that all those other cases are not true. The only one that holds true is an autosomal dominant inheritance pattern. The other interesting aspect of Creutzfeldt-Jacobs was that it was known to have a modified version of the prion gene that made it resistant to proteases, and they designate the nomenclature in the paper as a capital P, a small r, and a capital P, and that stands for protease-resistant prion protein. So in figure 2, what the group wanted to do was try to figure out whether individuals in this family that died of fatal familial insomnia also have a protease-resistant form of prion. So they carried out something that's called a dot blot, which is similar to a Western blot, except you don't run a gel. And they had 2 individuals that died of fatal familial insomnia, and they've designated those individuals as 4, 37, and 5. That's for their generation, 58. And then they have a control individual, which is brain tissue from someone who passed away of some other disease, but not fatal familial insomnia, not Creutzfeldt-Jacobs disease, or any other form of prion disorder. And they have a piece of nitrocellulose, which we said is just a fancy piece of paper. And they have 3 individuals that died of Creutzfeldt-Jacobs disease. So these are the different tissues that they had. Now, it turns out when they dissected the brains of the individuals that died of fatal familial insomnia and of this person, they took different regions of the brain that included basal ganglia, thalamus, cerebellum, and so on. And they have letter designations for each of those. So they have B for basal ganglia, for instance, and TH for thalamus, C for cerebellum, and so on. So there's a bunch of these. I'll just write them down here. You can take a look at the paper. So just different parts of the brain. And what they did is they just took that a small piece of brain tissue and they ground it up in a buffer solution. So this one would be basal ganglia, and the next one would be thalamus, for instance. And they grind it up. So they make their milkshake. But then they add protease K, which is a protease, as its name suggests, and proteases dissolve proteins. So it should, under normal circumstances, chew up all of the protein in the sample. And if you look at the control here, well, first of all, after they grind this up and add the protease, they then take a pipette tip and they just put a little bit of this on this piece of nitrocellulose. So some for basal ganglia, thalamus, and so on. Now, it turns out that you can't see the protein, so I'm just going to erase this now. Okay. But it's on there, okay? And then they do a Western blot, they carry out the antibody staining procedure of the Western blot, or the dot blot in this case. So, they have a primary antibody which recognizes prion, and they say in the materials and methods section that they obtained that from Stanley Prusiner, so not surprising. And then they buy a secondary antibody which binds to the first and then is conjugated to something that changes color. So you incubate the Western blot first with a solution containing the primary antibody that finds the prion protein if it exists. And the only way that it exists is if it's resistant to this protease, because otherwise it would be dissolved. And then you wash the blot a little bit and add the secondary that binds to the first, and then you add a little developer solution which causes the secondary antibody to change color. And if you look at the dot blot in figure 2, you can see that they have 3 different Creutzfeldt-Jacobs individuals, 3 people that died of CJD, and you get a nice staining for protease-resistant prion. And then if you look at the control individual here, in this region, you don't see anything for any of the different brain preparations, because all of the prion protein in that individual is normal and it's not protease resistant, so it gets chewed up by the proteases. But now if you look at the brain samples from the 2 individuals that died of fatal familial insomnia, you can see that they get some staining, basal ganglia, thalamus. This individual has some in thalamus, and some in this sample, and so on. They have this protease-resistant form of the molecule. So that provided them some evidence that individuals with fatal familial insomnia have this protease-resistant form of prion. So at this point they decided to do a real Western blot, and let me start with a new slate, a clean slate. And you'll recall that a Western blot starts by running a protein gel. So they have a gel, and the gel has these little wells in it, like this. And so on. And you fill the well with your sample of proteins. And in this case, they want to show both treated and untreated samples. So this one happens to be the control. The first one that they look at is the control individual, and they prepare a sample from the control, right? A little buffer, some protein, grind it up and make a milkshake, and then they have one test tube that they add the proteinase K2, so they designate that with a plus, and without the protease they have a minus, so they add it down here at the bottom of the gel. So they have a minus, that's without, and this is with. And they load other samples. The next sample is of individuals that have Creutzfeldt-Jacobs disease, and then finally they have some with fatal familial insomnia. And of course, you guys know that they run the gel and they get a banding pattern, which I'm not going to draw in because it's invisible unless you stain it. But you guys know that you end up getting a banding pattern, and each band represents a different protein, and the small proteins run faster. So the smaller stuff is down here and the bigger stuff is further up the gel. And then after they run the gel, they transfer that banding pattern to, once again, nitrocellulose, fancy paper, and they run the electricity in the gel in this direction, and then they run the electricity through the gel so that the proteins get scared out of the gel and bind to the piece of paper behind it, and then they just throw the gel away. So in the end you just have this piece of paper. Now, they did these experiments at different time points, and all they show you is a strip of the piece of paper, like one lane. They show you this lane here like this. So they're cut out, and the first lane that they show you is again the control. So it's the control, and it's without the protease. So what they end up getting are these two pretty wide bands. I don't know why there's two bands, but that's the banding pattern that you see when it's stained with the antibodies again. So it's the primary against prion, and then a secondary that binds to the first and changes color. And then they have a lane where they add the protease, and you don't see anything because all of the protein gets ground up by proteinase K. Then they show you the three different individuals that died of Creutzfeldt-Jacobs disease. So they only show you one without the protease. So they have minus. So this is individual number 1 of Creutzfeldt-Jacobs disease. And then this is individual 1 with the protease. And what you see here is that you get a similar banding pattern as the control individual in the absence of the protease. But then when you add the protease, you do see some bands because it's protease resistant, and you get two smaller bands. So the big stuff is up here, and the smaller stuff is down here. So there's a change, there's a shift in size and their ability to migrate in the gel. And I think that this is because the proteins must bind to one another and form aggregates. So if you have some prion proteins like this, the core here is protected against the protease, but the protease could cleave little pieces of the prion that stick out from the central core, and it makes this aggregate smaller. I mean, smaller in size because you're kind of clipping away at the ends that are sticking out. And that also explains why Stanley Prusner said in his paper that prion was in every single sample that they tested as they took the sample through the biochemical preparations. Because if you have something like, the test tube with the different sucrose fractions, or if it's a continuous fraction that we talked about, you might see this as well. But, small aggregates could be in the 12%, and larger aggregates could be in the 15%, and really large aggregates could be in the 45%, and individual monomers could be in the supernatant. So, at any rate, you get this shift in migration, and you still see protein after the digestion because they are protease resistant. And if you look at the second person and the third person that died of Creutzfeldt-Jacobs, then you see a shifting pattern. They're different actually if you look at it, but still there's a shift and they're smaller than the original here or here. Then they look at the individual that died of fatal familial insomnia, so 437 for instance. This is with, and this is with, this is without, and this is with. And without, they show a big smear up here again. And with, you get a shift. And then if you look at the other patient, which I'm not going to draw here, I ran out of room, but you see the exact same, pretty much, I mean, not exactly the same, but you see the same phenomena of a large smear up here of the protein, but without the protease, and a nice band here with the protease. So it showed that indeed individuals that died of fatal familial insomnia have this, again, protease resistant form, and I think this example that I drew outlines how you get the shift in the migration in the gel. Based on the pedigree, the scientists knew that this was an inherited disorder, that it wasn't sporadic, and it must be caused by a mutation in a gene. Since individuals with fatal familial insomnia have this protease-resistant form of prion, the scientists suspected that the mutation might be in the prion gene. The coding sequence for wild-type prion had been sequenced before, so it was available in PubMed. And they could look this sequence up and generate PCR primers based on that sequence that would allow them to amplify the coding sequence of prion in individuals of this family afflicted with fatal familial insomnia. So if you have an RNA molecule with a poly A tail, we've said before that in order to sequence the molecule, you have to convert this RNA into double-stranded DNA that's called copy DNA, or sometimes they refer to it as cDNA. And to make that conversion, you have to bind oligo-DT, which is a string of T's that bind to the poly A tail of the RNA molecule. And you add reverse transcriptase, which makes a DNA copy, a DNA complementary strand to the RNA molecule. So this bottom strand is now DNA, and the top strand is RNA. Then you add an RNase, which chews up RNA, and it gets rid of the top strand, which I'm not going to erase. I'm going to leave this here. And then you add DNA polymerase, which makes a complement of the complement. So you generate a double-stranded DNA molecule in which the top DNA strand is identical to the RNA molecule, except that there are no U's, it's T's now. We said in PubMed that they only publish one of the two strands because PubMed knows that if you know one of the two strands, you can easily deduce the second strand because it's just the complement. And the top strand is the copy, the DNA copy of the RNA molecule. And if you translate that sequence into all 6 reading frames and you look for the longest open reading frame, you can then find the start site, which of course is the codon ATG that encodes the first methionine, and you can also find the stop of that longest open reading frame. And if you know this sequence from PubMed, you can simply design PCR primers that allow you to amplify the RNA from individuals that died of fatal familial insomnia based on this published sequence by making a primer 5', this is the 5' end of this top molecule, this is the 3' end of the bottom, and this is the 3' of the top and the 5' of the bottom. But you can make a primer that's just on the outside of the start site, which will bind to this complementary strand, and then make a second primer which is just after the start site. And if you isolate RNA molecules from brain tissue of individuals that died of fatal familial insomnia, you purify the RNA and convert it into double-stranded DNA, all of it, all of the different RNAs, and then use these PCR primers and the reagents needed to carry out PCR. You will then amplify a stretch of the RNA molecule, now converted into double-stranded DNA, from the end of this PCR primer to the end of this PCR primer. And that will contain the entire coding sequence. So they carry that experiment out and they find that individuals that died of fatal familial insomnia indeed have a single point mutation at codon 178, which converts the codon from GAC to AAC. So this first nucleotide is altered, and that converts the amino acid from asparagine to aspartic acid. Okay, you guys, I realize I misspoke about this primer. I said earlier that this primer was after the start site, but obviously it's not after the start site, it's after the stop site. So sorry about that mistake. They say in the paper that this mutation abolishes a restriction enzyme site. Now look, we talked about restriction enzymes before. We said that it cuts double-stranded DNA at a very specific location, and it turns out that if you analyze the wild type version of prion, that a specific restriction enzyme that's called TTH111, that's the name of the restriction enzyme, that if you added this enzyme to a PCR product of wild type prion coding sequence, that you would cut the DNA. And in individuals that have the mutation, their version would not be cut. So they use this as a test to determine who in the family has the mutation and who doesn't, and whether all of the individuals that have the mutation have the disease. So they show you a gel of a PCR reaction, and they have different individuals that they've looked at. Individual 1, 2, 3, 4, 5, 6, and then they have a molecular weight marker, which is just DNA fragments of known size so that you can determine the size of your PCR products. So if you amplified an individual that had the mutation, they should have one copy of the mutant allele and one copy of the wild type allele. So it turns out that the distance between these two PCR primers from here to here is 800 base pairs long. And an individual that has the mutation will have one band that's 800 base pairs long. So here, 800. Then that individual should also have a wild type form of the gene, and the wild type form contains the restriction enzyme site, and it cleaves the DNA such that one fragment is 556 base pairs long, and the other fragment is 244 base pairs long. So they have one at 556 and 244. So if you look at figure 5 at the gel, you can see that individuals 1, 2, and 3, which by the way are all afflicted with fatal familial insomnia, all have the 800 base pair band, which does not have the restriction site. So they just get one band that's 800 base pairs long, and then in those individuals they also have a band that's 556 base pairs long and one that's 244 base pairs long. Now in my copy of the paper you really can't see the 244 base pair band, but it's there. It's just faint because it's small. In the individuals that are wild type, which are 4, 5, and 6, they do not have the 800 base pair band, which is indicative of the mutation. They just have copies of the 556 and the 244. And obviously 556 and 244 add up to 800 base pairs, which is the overall length of this molecule. They also say in the paper that they've found individuals that have the mutation in the family that's of the right age, older in life, to have the disease, and they don't have it. And that suggests that there's incomplete penetrance. That is, not every single person that has the diseased allele gets the disease. And we've also said that if it's an autosomal dominant disorder, that the diseased allele must be expressed because that allele is making you sick. If you just had one bad allele that wasn't making the protein, the good copy would make enough of the protein so that you'd remain healthy. It's the fact that you're making a protein that has a mutation that doesn't function correctly that actually makes the person sick and causes the disease. So it looks like prion and Alzheimer's, for that matter, are caused, the genetic forms are caused by mutations in different disease genes, and it looks like the proteins in both cases form aggregates, and somehow these aggregates lead to these different neurological disorders. Okay guys, that's it for today. Again, I hope you're all well, and I'll talk to you tomorrow. Bye-bye.